Hi, everyone. So today I will talk about the research that I started here as a research fellow just over a year ago on the evolution of the bacterial flagellum. So I will start my presentation just giving a brief introduction about myself, so you know a little bit about my background and how I got here. And I will also talk about why phylogenies are so important in biology. And I am aware that for many of you, this is very redundant, but for some of you might not. And the challenges that we face when we are trying to infer deep phylogenies, especially which is the case for very ancient biological systems, such as the bacterial flagellum. And I will present our work on the phylogenetics of the flagellar proteins and how we're incorporating protein structure data into our approach. And then I would end my talk talking about what we've been playing on how to potentially build phylogenies using protein structure data. So of course, the work that I would present here, I haven't done it on my own. I've been working with Nick and Jordan here at the University of Auckland. And we are also collaborating with the University of New South Wales and Wisconsin in the US. So I am originally from Colombia, where I did my bachelor's in biology. I then did my PhD on plant phylogenetics in Australia. So my background is in plant phylogenetics and biogeography. I then went to the US and I worked on the DNA barcode project at the Smithsonian. And there we were using DNA barcodes to build phylogenies, mostly for conservation for biodiversity assessments. And then I did a fellowship at the US Food and Drug Administration, where I used DNA barcodes and phylogenies to develop qPCR methods to identify plant allergens in food products. Then I took a break for parental leave, and now I started again in phylogenetics here with this project. So throughout my career, I have used phylogenies in very many different ways. And I am very aware of how useful phylogenies are in very different fields. So for example, in systematics, we obviously use phylogenies to organize our knowledge of biological diversity. Taxonomy is often based on phylogenies as we aim to reflect evolutionary relationships when we name organisms. And they also provide insight into past evolutionary events at all levels of diversity. So not only a species level or genome, but also genes, and in this case, protein level. And having this knowledge of past evolutionary events is really useful to make some predictions about what we can expect, especially, for example, in conservation. So we are all familiar with phylogenics and forensics, in epidemiology, systematics, and sometimes we develop methods in phylogenetics that are then used in other fields of biological research. But, and to me, the most important things about phylogenies is that first, they are interesting by themselves, but they underpin and they provide the framework to any biological research question, and that is the evolutionary theory. Phylogenetics provide that framework in which every question we have in biology has to have this base. It just makes sense when you look at it with a phylogeny, to me anyways. So phylogenies are not trivial to build. There is a lot of methodological and philosophical challenges, and some of them are often or can be exacerbated depending on what level of the phylogeny you are. So if you are in the shallow branches of the phylogeny, you might have some level of confidence that the variation that you see in your data, which is often DNA and or protein data, is really reflecting the common ancestry between these species or proteins. I'm going to talk about proteins because this is for proteins. But as you go deeper in your phylogeny and further back in time, So when you're not talking about millions of years, but billions of years, then there is a lot of variation in your data, and a lot of this variation does not reflect a common evolutionary history, so we call it noise, it's noisy. And so your phylogenetic signal weakens as further as you go here. And this is a very big problem when you're trying to study ancient systems, such as bacterial motility. So bacteria have been moving since before there were bacteria. So the last bacterial common ancestor was already model. We know that bacteria move in very different ways. They can swim, they can glide, they can swim, they can do all sorts of things. This is a very, very ancient adaptation. I am interested in one specific type of motility, one that was already present in the last common ancestor of bacteria, and this is the bacterial flagellum. So this is a flagellum. It's like a little tail-like organelle that is used for motility. The flagellum rotates at very high speeds to propel bacteria through liquid environments. And this really plays a very important role on how bacteria disperse, how they avoid immune response, or how they form biofilms, among other processes. So the bacterial flagellum is not only very ancient, but it's also very complex. It is made of roughly 30 to 40 proteins, depending on the bacteria group. And these proteins self-assemble into 3 modules. You have the basal body here. You have the hook just here. And the hook connects the basal body to the filament. And the filament is the tail that you can see outside the cell. For the ones who have a flagellum outside, because you have some bacteria that have the flagellum inside, which is another story. So the flagellum is very complex, and we really like to call it a classic example of a complex evolutionary adaptation. And we do know very well how it works. We know how these proteins work together to create motility. But we still do not know how this very complex system originated and evolved. And there is a group of people called the creationists in the US that also love the flagellum because they claim that it's a great example of intelligent design. And this is how Nick, my boss, got into this. He wanted to fight creationists, and he's still doing it. So we have this project. And according to them, the flagellum is a great example because it's a functional unit. It cannot work separately. And you cannot explain the existence of a functional unit through gradual variation and natural selection. Based on the evolutionary theory, we can explain the origin of the flagellum as a modular system that came together by borrowing different parts of different proteins and putting them together into this system. And we can test this hypothesis by looking at the homology of each of these proteins that make the flagellum. And this is not new. This has already been done for one of them. So this is a type 3 secretion system. So this is the basal body of the flagellum, and this is the filament. And at the base, on the cytoplasm side of the basal body, you have a type 3 secretion system, which is pretty much an ATPase. ATPases are very old proteins. And this type 3 secretion system has homologues in all of the 3 domains of life, in archaea, in bacteria, and in eukaryotes. And very interestingly, there is very strong evidence that these homologues actually derived from the ATPase from the flagellum. So this is telling us that the flagellum might be that old, even before. But most importantly for this case is that this is evidence that you take one of the proteins in the flagellum and put it in a different structure, and it still functions. It's still doing the job. It does not need any of the other flagellar proteins to do what it needs to do. Then, so I think we have evidence that the flagellum, it is a modular system. Now the question is, well, how did all these proteins come together? This is a question that I hope I will answer during my lifetime. And for that, we are building phylogenies for each of the proteins that make the flagellum. And once we have a phylogeny for each of the proteins that make the flagellum, we will test for congruence of a common history, and hopefully we can put a picture of the major stages of flagellar evolution. And then we would have a nice framework to finally address the question of how these proteins came together and how are they co-evolving for billions of years. To do this, I am using a broad range of methods. So I'm doing remote homology searches. I'm using full genomes. I'm looking at gene order. We are also looking into different protein substitution models that take into account that these proteins are very ancient and have accumulated a lot of variation throughout billions of years to build phylogenies using maximum likelihood and Bayesian approaches. Then we're using protein structure to characterize the flagellar and the non-flagellar homologues. And to do this, we need to look at the fold and domain structure for each of these proteins and the domains that make these proteins. So where do I start? So I have started with the stator motor proteins. So this is an electron microscopy image of the basal body. And surrounding the basal body, you have the stator motor proteins. There are two proteins. This is mod A and mod B, and they have been named like that after E. coli. So mod A is a pentamer that works as an ion channel, and mod B is a dimer that works as a plug to regulate the ion flow through the channel. So the function of these proteins is to provide the energy for the rotor to rotate and generate the torque for the filament to move. And they do that by creating a proton gradient across the membrane. Some bacteria use protons, and some would use sodium, and some would use both. What do we know about these proteins? We do know something about the structures, where we know they're pentamers, and we know these are dimers. We know that mod A belongs to a family of proteins that comprises, they're all proton channel proteins, they're all membrane proteins, and the best known homologues, so TOLQ and XBBB, they use this energy for active transport of large molecules across the cell membrane. And for mod B, we do know the homologues. So we do know TOL-R works with TOL-Q, and XBBB works with XBBD, and this is all named again in E. coli. And all of these proteins, again, act as a plug to prevent or to regulate the ion flow through the channel. One thing about mod B is that we do know these are structural homologues, but sequence similarity has been lost. So when you put these sequences together, they don't look very alike. It's very hard to align them, which is important from what I'm doing. So this is a phylogeny of mod A. To build this phylogeny, I took about 180 bacterial genomes, and I took the mod A sequence for E. coli, and I did a remote homology search, and the output was about 500 homologues across 140 taxa. So these proteins are actually seem to be very common across different lineages of bacteria, whether they are motile or non-motile. We also found that some of these homologues have multiple copies within the same taxon, or that one taxon has multiple homologues. And we also detected some proteins that are involved in other types of motilities, which is gliding motility. So all of the proteins in this phylogeny are pentamers, ion channels, with a plug in the middle to regulate ion flow. There are two main groups. One includes all of the flagellar proteins, so all of the mod A and alike. And there is a very large group that includes non-flagellar proteins. So these are proteins that are using ion flow to transport large molecules across the membrane, or to do something that we still do not know. So we have this very large group of proteins that have been annotated, and this is based on automatic annotations that we do know they do have a homologous domain to mod A, and that they belong to this family, and based on structural data that I will show, that they are also pentamers. So presumably they're also ion channels, but we do not know to what system or what are they doing. So this is the mod A phylogeny. To build the mod B phylogeny, if you remember, I did homology searches using sequences, but because there is very little sequence similarity between mod B and its homologues, it was very unsuccessful. So to build a mod B phylogeny, I used gene order. So mod A and mod B have been working together for a very long time. So you can expect them that they would be in the same operon in the bacterial genome. So we took each of them, and we searched for the gene, for the mod A gene in each genome, and look at the gene next to it. And we expected that would be mod B or just the plug. And yes, it was. So for mod B, at each time we found mod B, and the same for the homologues. And even for the AQB proteins, that we call them like that because we don't really know how to call them, we found that there was a small protein, or about 100 amino acids, that had been annotated as either mod B or its homologues, or a protein of unknown function. But again, by looking at the structure, it does look very much like a plug protein. And this is how we built the mod B phylogeny. So here you have the mod A phylogeny, and this is the mod B phylogeny. And as we expected it, they are very congruent. So these proteins are working together, so they are co-evolving. And you also have a flagellar clade for mod B and a non-flagellar clade for mod B. And here, the colors of each clade is, so they're colored by the channel, which is here. And then it just shows where they correspond. And if they are the same color, then it means the protein, it is actually downstream in the genome. And so this is still a working process, but something is very clear, and the flagellar clade is just very clearly and very consistent that these proteins are working together. We also saw systematic differences between the flagellar and the non-flagellar clade when we looked at the sequences. So this is only the mod A phylogeny, and this is the core region of the mod A alignment. And here you can see, well, easily what is determining the two groups in the phylogeny. So you have a group of proteins here on top that have residues at both ends, right? And then you have a group of proteins that don't. So all of the proteins that belong to a non-flagellar clade lack these residues here. And all of the proteins so far that we have sampled that have these residues here, they all belong to the mod A clade, to the flagellar clade. And within the flagellar clade, you have two also very defined groups. One that you can call it Crassus mod A, but really we all call it chunky, because the first time I was trying to explain what I was seeing to my collaborators, I was just, oh, there is this chunk here, and the chunk kind of just stayed. So I'm trying to call it Crassus, but really we just call it a chunk. So we have a chunky mod A and a non-chunky mod A. And again, this is very consistent in all the proteins we look at. So the question was, okay, we see a clear pattern in the sequence data. Are these sequence differences in the sequence, how will they translate into the structure of the proteins? What would they look like? So to do this, we are using protein structure predictions using AlphaFold. So for the ones of you who are not familiar, you pretty much give AlphaFold a protein sequence that it compares using a multiple sequence alignment, and it uses this information to pretty much give you a protein structure in about less than an hour. This is something that if you go to the lab and try to do this, it's very tedious. But here now we can do it very fast. So we can now have protein structures for all of our proteins. So this is the work of Pietro, who's in Sydney. And he's been really looking at these proteins a lot. So these are the mod A subunits. So this is the ion channel here, and you can see the plug, the mod B here in the middle. So the A subunits have two layers. They have the inner shell, which is the side of the protein that interacts with mod B. And then you have an outer shell that is the one that interacts with either the cytoplasm or the membrane. And here are the proteins and how they are positioned in the cell. So this is the membrane, the cell membrane here. So this is the part of the protein that is embedded in the cell. This large part of the channel is in the cytoplasm, and there is a tiny bit that comes in the periplasm. And here, each part is colored, whether it is in the outer shell or the inner shell. And one of the traits that is very easy to look at, like you don't have to be a structural biologist, I don't think, you can see this square. This is what I call the mod A square. And all of the flagellar proteins have this square. And it is only present in the flagellar proteins. And the non-flagellar proteins have they don't really have a shape, but they kind of just have that. And here you can see that also in the domain that is in the cytoplasm, you also have a very unique feature that looks like this. So this is the chunky mod A. So chunky mod A actually looks like a little loop, and the non-chunky mod A looks like this. And you only have this in the flagellar clade. And then when you go outside the flagellar clade, you find much more diversity on the shape of these proteins and what they look like. And also very interesting is that the flagellar proteins do not really have any periplasmic domain, whereas the non-flagellar ones do. And some of them have really large and crazy looking ones. And these are specific to those AQB proteins, the ones that we don't know what they're doing. So the next thing we're going to do is to only take those periplasmic domains and try to do some deep homology searches to try to find out what function these domains have. So we can at least have a better idea of how these AQB proteins, what are they doing? So the nice thing about this result is that you don't have to take your phylogeny. You don't have to build a protein phylogeny to be able to predict to which group the protein belongs to, because we have structural traits that identify the protein for you. So this is for me, it's also very nice evidence that the groups that have been inferred in our phylogeny, they are supported by this. They are supported by structure, which is nice to see. Also, the chunk, the crassus mod A. So Pietro is very interested in seeing, okay, there's this chunky thing, this loop. So is it affecting the way these proteins are interacting? So he found the YCGR protein, which is a protein that has been reported to express only when bacteria are, for example, in biofilms. So when they do not need to move anymore. So when the bacterium is not moving, YCGR comes here and inhibits the flagellar system. So it stops rotating. So he's been doing, and these are very preliminary AlphaFold simulations. So this needs to be tested in the lab and in other ways. But what he has found so far is that the chunky mod A and the non-chunky mod A do not interact the same way with this protein, and that actually the chunk seems to like it more. Seems the YCGR seems to like the chunk more than the non-chunky ones. We don't know how to explain that yet, but that's where we are at. And again, this is a way of, we can now understand what this could potentially mean in the future, just because we have a phylogeny. So we were able to find this because we have a phylogenetic framework. So we kind of knew where to look at and what to look for. Okay, so I want to finish my talk with something that we have been playing recently. And it's about using protein structure data, not to just inform alignments or to interpret phylogenies, but to build phylogenies, which a few years ago would have been fully impossible. But now, because we have AlphaFold, seems to be possible. So why? Why would we build phylogenies using structure? Well, because protein sequences, as mod B is a really good example. They can be highly divergent. Sometimes they are hard to align, and if you don't have a good alignment for which you are not confident of your assumption of homology, then your phylogenies become a little meaningless. But protein structure is very much more conserved than sequence, which makes it a very unique and powerful source of information to infer deep phylogenies. And also, if we better understand the structure of proteins, then we can better understand the function of proteins because they are very strongly correlated. If we can better understand the evolutionary history of the function of these proteins, then we would have a better chance of putting together the flagellum puzzle. So what we're trying to do here is that we are using FoldSeq. FoldSeq is a tool for structure-based homology search in proteins. And pretty much this is the workflow that they show in the paper. What you get here is that what FoldSeq does is that you give it a query protein and it has a bunch of target proteins. And based on this, it learns, it takes the interactions between the residues and it determines the position of the residues. And by doing that, and it discretized each residue, the position of the residue, it puts it in what they are calling a structural alphabet. So FoldSeq is not coding the position of the residue. It's taking the backbone of the protein, it's taking the residues in the backbone of the protein and coding the interactions between the residues. And by doing this, it's actually capturing the most conserved properties of the protein, which is what we want. And well, just want to go rapidly to this. And the way it's doing that is that it gives a virtual center to each residue, and it determines the position of each of the residues based on the position of the virtual center. And by residue, I mean amino acid, each amino acid. And here is a way of how it's plotting this. So each circle here represents a residue and is colored based on the nearest centroid. So this one here. And all of this information is discretized into what they're calling, and it puts each residue in a state. So there are 20 possible states for each amino acid. And it builds an alphabet assigning one state to one amino acid. And the result is something that looks like an amino acid sequence, but it's not an amino acid sequence. It's an 3DI alphabet. So here, each letter represents a 3DI state. And one of the things about this is that when you look at the sequence identity in the same proteins, only using amino acids, for this protein in particular, you would have 10%. But if you use the 3DI alphabet, then the sequence identity doubles, which is very significant. So this is just an example of how an alignment of the mod A proteins would look like, an alignment of the 3DI alphabet for the mod A proteins. And it just looks like something you can align. And here it's also very interesting. And again, we're just really in the proof of concept step. So this is still very early. We still have a lot of things to figure out. But so far we've seen, this is what it looks like. And here on top, if you remember, the mod A had these very additional residues. It seems that when you take the 3DI alphabet, first, you have some states that are very conserved across proteins, just as amino acids. So this means that these positions of these residues, again, this is based on only 10 sequences, would be more conserved than other regions here. But you would also see that since it's assigning a state for each residue, you can also identify that these, for example, are the flagellar proteins would have additional states, would have like chunky states. And then just as a proof of concept to play with, We build a phylogeny. We put this on IQ3, see what happens. And it does give you a phylogeny. So yes, it is possible. It is possible to build a phylogeny using structural alphabet. But there are a few things we still need to consider. First, how are we going to align these these structural states? And for this, FoldSeek actually does this as part of the homology search. It's aligning 3D alphabets to be able to determine what are the most similar proteins. So we're trying to do is also to borrow the way they are aligning the 3D alphabet to create an alignment to build a phylogeny. And then also, when we're building a phylogeny, well, if we're using model-based approaches, what would be the best model that would best suit this type of data? So we still have a few things to figure out, but it is possible. And I obviously want to thank my collaborators, and I do want to advertise once more that we're organizing an Australian structural phylogenetics meeting sponsored by CCE. And it will take place in October. And if you're interested, please follow the link and register. We still have some spots left.